For decades, computational models of respiration operated under a structural flaw. They treated the neural pattern generator in the brain and the pulmonary gas exchange in the lungs as isolated, unresponsive silos. Legacy models typically stop at eupnea. They focus entirely on quiet, passive breathing, where ventilation is driven solely by the rhythmic contraction of the diaphragm. But mammalian physiology possesses a built-in anomaly. Under extreme metabolic stress, such as severe hypercapnia, the system abandons this passive rhythm. It physically transitions to active expiration by engaging a completely separate secondary neural oscillator. This schematic from Molkov and colleagues maps out the entire closed-loop control system. By mapping the three-part connection between the brainstem network, the spinal cord, and the lungs, we can trace exactly how chemical and mechanical feedback force the physiological transition to active expiration. Decoding this transition identifies the mammalian brainstem as a reactive system, shifting its architectural state in direct response to shifting mechanical and chemical feedback. To understand quiet breathing's failure point, we isolate its neural engine, the ventral respiratory column. Here, early I, post-I, and aug-E neurons trade signals, regulating the inspiratory cycle. This rhythm feeds premotor output. The ramp-I neuron sends excitatory drive, instructing the diaphragm to draw air in. In this baseline state, expiration requires no neural command. It is entirely passive, relying on the physical recoil of lung tissue to push air out. This passive VRC oscillator is efficient, but it operates blindly. Without physical feedback from the expanding lungs, the neural engine would continue to contract the diaphragm, risking severe tissue overinflation. The brainstem requires a sensory mechanism to know exactly when the lungs reach their physical capacity. This data is gathered by pulmonary stretch receptors in the lung tissue. As lungs expand, they measure physical stretch and transmit signals up the vagus nerve. This triggers the Herring-Brewer reflex, sending an inhibitory spike to forcibly override the rhythm and terminate the breath early. This chart proves the necessity of that reflex. Panel B models a vagotomy, severing the stretch receptor pathway. Without mechanical feedback to cut the inspiration short, the ramp-I firing durations widen dramatically, and the total lung volume spikes to dangerous maximal levels. Mechanical feedback perfectly governs the physical timing of each breath. However, it is entirely blind to the chemical composition of the blood. A metabolic crisis introduces a problem stretch receptors cannot solve. As a mammal enters progressive hypercapnia, carbon dioxide levels in the blood surge. Simply adjusting the timing of diaphragm contractions is no longer sufficient to clear the toxic gas. The brainstem tracks this DRET using the retrotrapezoid nucleus, or RTN. At critical CO2 thresholds, the RTN injects excitatory drive to awaken the dormant late E neuron. Instead of routing through the diaphragm, it fires down the abdominal nerve, triggering abdominal muscles to contract upward. To solve a chemical imbalance the diaphragm cannot handle, the brainstem physically recruits a completely independent expiratory muscle group to force air out. This timeline tracks the onset of progressive hypercapnia. It reveals a complex synchronization problem. The secondary abdominal pump cannot fire randomly, or its forced exhalation would physically clash with the diaphragm. The brainstem prevents structural collision using heavy inhibitory gating. The late E neurons are suppressed by the primary inspiratory network, restricting their ability to fire exclusively to the very end of the expiratory window. As CO2 concentrations rise, the late E neuron overrides this suppression through quantal acceleration. The secondary oscillator breaks through the inhibition, firing in strict integer ratios. At the first breakthrough threshold, the chemical drive allows the late E neuron to fire only once for every 3 primary phrenic bursts. As hypercapnia deepens and the blood becomes more toxic, the chemical drive pushing from the RTN intensifies, breaking through the inhibition more frequently. The firing pattern accelerates, quickly locking into a precise 1:2 ratio. Finally, the chemical emergency reaches its systemic climax. The excitatory drive from the RTN completely overwhelms the intermittent neural inhibition. The system achieves a 1-to-1 phase lock. Every single diaphragm contraction is now immediately preceded by a forced abdominal exhalation. Quantal acceleration provides an elegant mathematical solution. It seamlessly merges a secondary explosive motor rhythm into the primary breathing cycle without disrupting the baseline architecture. What is the actual physiological payoff of activating this secondary oscillator? This graph tracks ventilation as CO2 rises. Notice the blue line for respiratory frequency. Crossing the active expiration threshold, the vertical line, the rate stays flat. The lungs don't pant faster. Instead, the teal tidal volume line surges upward, instantly increasing breath depth. The abdominal pump does not expand the lungs. It physically squeezes out the functional residual capacity, the pocket of stale air normally left resting at the bottom of a passive exhalation. Under severe metabolic stress, simply accelerating the breathing rate yields diminishing returns. To guarantee survival, the system structurally alters the lungs' resting baseline geometry, creating a larger empty envelope to move significantly more volume per stroke. Mammalian respiration relies on a nested hierarchy of chemical and mechanical thresholds. Mechanical feedback optimizes the physical geometry of quiet breathing to prevent tissue damage, while chemical feedback recruits the forced expiration circuit precisely when those mechanical limits reach their ceiling. This reveals why computationally linking abstract neural networks directly to physical tissue models is required to decode mammalian biology. You cannot understand the software without mapping the physical constraints of the hardware. The brainstem acts as a coordinator of physical and neural systems, calculating precise changes in lung geometry to sustain gas homeostasis.